Good morning to everybody. I would like to thank the organizer for giving me the opportunity to participate to this meeting, in particular Dr. Alagabr. And what I'm talking about today is we are studying the molecular mechanism for stemness. So we use the cells just as a model to understand how they work and how they can maintain their stemness. I don't know, okay. In particular, we focused our attention on the microRNA. MicroRNA are small RNA that act as regulatory. And they inhibit the translation of protein and are implicated in different processes, such as, I don't know the pointer. Okay. Okay, you can see. There are okay, no. Okay, doesn't matter. Don't worry. Ah, the mouse. Okay, they are involved in different process for regulation of apoptosis, survival, and replication. So we focus on, in particular, for heart disease, and we use cardiac stem cell as a model to understand better how we can use microRNA and how microRNA are involved in the reparative process in cardiac diseases. Okay, sorry. Okay, our model consists in the isolation of so-called cardiac stem cell or cardiac progenitors. In vitro, we can take the biopsies and try to generate for different passages cardiosphere or cardiosphere-derived cells. These are so-called progenitor for cardiac disease. They have been used in different clinical trials, and we try to understand which is the pattern of microRNA expression in the cells. For doing that, we use microarray analysis and next generation sequencing analysis. As you can see, this is the result for the heat map for microarray analysis. You can see that the biopsies clusterize in correlation with the spheres and the CDCs. So you can see that the expression of microRNA for biopsies is similar for different patients and is different from the most undifferentiated progenitors. The same result is, these are results in preparation, is not paper yet. The same result we got from the UP gene analyzed by next generation sequencing. So you can see that biopsies have a pattern that is similar among them and is different from the one that we see in the most undifferentiated progenitor that we isolated. The same for the gene down. We analyzed by the Diana tools this kind of pattern, and we see that the gene that have been identified are involved in different processes. This is the upper gene, the microRNA that are upregulated in progenitor and undifferentiated progenitor. These are the genes that are upregulated in biopsies. So we try to put this gene and to understand which gene with this microRNA and trying to understand which gene are regulated. With the tools, we found a different pathway that are involved in cardiac repair, in pluripotency, and another pathway that is EPO pathway, that is a regulator of size of organ. What we are going to do? We just confirmed by real-time PCR this gene. Some of them are upregulated in biopsy and down-modulated in most of undifferentiated progenitor. Some of them are upregulated in undifferentiated progenitor. We try to make a correlation either with the age. The genes that are upregulated in undifferentiated promoter, in undifferentiated cells, are down-modulating during the age of the patients where it come the biopsies. Each dot is the patient's biopsies. In the opposite way, the genes that are down-regulated in the most undifferentiated progenitor going up with the age. So there is a direct correlation with the age in the expression of in undifferentiated gene cells. What we can do next? We can try to inhibit the gene that are involved in this process, or to mimic the expression of microRNA. To do that, we use the technology that is called a small molecule that is called LNA, that is locked nucleic acid. This particular nucleic acid has a chance to enter directly into the cell without any carrier by gymnosis, and was used in different trials, and is able to bind specifically with high affinity the microRNA or to be used as mimic. In this particular experiments, we used the LNA as inhibitor of microRNA. In particular, we used it against microRNA 34 that was demonstrated to be a barrier for somatic reprogramming, and was used in cardiac aging in mice. So if you inject in the mice, in this paper, they injected the LNA inhibitor against MIR34, and they get an increase on the myocardial infarcted mice, an increase of muscle replacement. To understand the mechanism, how it works, we did the same in vitro on cardiac stem cell. First of all, we tried to understand if the cardiac stem cell human cardiac stem cell contain this microRNA, and we isolated the slow dividing cell among this population, and we make by CSFA is living dye marker. We check the expression of microRNA in the low dividing cells, that is CFA plus, and you see MIR34A is highly expressed in low dividing. So we can try to inhibit this microRNA in these cells because it's present. We correlate this microRNA either, as we did, with the age, and there is an increase of this microRNA with the age of the patient in the biopsies. So if we give Antagomir, the inhibitor of MIR34, we have an increase of growth in vitro of these cells. Why we have this result? Because we have a change in the expression of some genes.